All right, so today we're talking about DNA. This is the start of our new unit. We'll be discussing essentially the whole cycle of a cell growing and copying its DNA and dividing into two new cells. Okay? But to start out with, let's just talk a bit about DNA. Does anybody know, like, what is DNA for? Like, what's the purpose of DNA? What's it do? Okay, yeah, it tells you, like, it's the code for what you are, like what you look like, right? Your phenotype, and a whole lot more than that too. But yeah, can we use DNA to tell the difference between individuals? Yeah, it's like a fingerprint, right? If I find some blood on a murder weapon, like, we can identify the DNA and we can pin it on a suspect, right? So we have differences in our DNA, obviously. And as Tommy hinted at, it is the code, it is the actual genetic code that makes us all unique individuals. Now, the differences in your DNA between, say, you and I, they're really not that many. There's not that many differences in our DNA, but we can still pick them out, like, precisely. In fact, you share a fair amount of DNA, an awful lot of DNA, with, say, the chimpanzee. Humongous amount of DNA you share with a chimpanzee, right? You share an awful lot of DNA with a banana. Well, I know it's strange, right? But it shouldn't be too surprising, because if you trace things all the way back through time, right, all of life on the planet is related. Like, everything is related. And there's no more striking example of that than looking at the DNA and how DNA is similar between different organisms, right? Now, like I said, we're all the same species. Our DNA is incredibly similar. We're talking about very small differences that enable us to fingerprint the difference between individuals. And oftentimes the differences are in pieces of non-coding DNA that don't actually code for a specific protein. But Tommy's right, like, the DNA is what gives me blonde hair. Right? Might give you red hair, might give you, dark colored hair, right? It gives us all those different characteristics. And it, in many ways, constrains what we are and who we are. Now, humans are a bit different than other organisms. We're thinking, we're conscious beings that can modify our behavior and our thought patterns, as well as our bodies, right? Anybody ever dye their hair? You're not stuck with what your DNA gave you. You can just go dye that stuff, right? And you can change your thought patterns. But in other ways, your DNA really constrains you. Like look at me, right? I could practice basketball my whole life. Am I going to be like a superstar basketball player? Probably not, right? No matter how good I get at basketball, I'm not going to be able to compete with those guys. I'm not that tall, right? It's not going to happen for me. I could practice football my whole life. I could probably get pretty good at football. I'm a pretty athletic guy. But those guys that play professional football, they're monsters. They're huge, right? I couldn't get that big. Like, I don't have that big bones. So your DNA does constrain you. And I know that we're taught from early on, like, you can do anything you like and be anything you like. And it is very true, right? Like, especially in America, we have so many opportunities and so many ways we can be and things we can do. Like, the sky is the limit in many ways. But we also have to respect the fact that, like, our DNA is the code. And, like, sometimes we've got what we've got, right? And we do what we do with what we've got. So DNA does have a lot of stuff to do with who we are. Like, it's just how it is, right? And chemically inside, things you can't even see, DNA has control over. So let's talk a little bit about DNA today and sort of how it does what it does. And I'd like you to write this down. The name of DNA, when you spread it out to all of its letters here, is deoxyribonucleic acid. And it says what it is right there. What is it? It's deoxyribose, so that's the name of the sugar that's in it, and it's found in the nucleus. How about that? Right? So this is the molecule that transfers our heredity. Does anybody know where their DNA came from? Where did you get your DNA? From where? From mommy and daddy, right? It came from mommy and daddy. That's where your DNA came from. And we know that today. We understand heredity. We know that the characteristics you have were passed down from your long family tree somewhere. Now I have up here that it's packed in chromosomes. I want to kind of qualify that because this is the case for eukaryotic creatures like ourselves and plants and fungi, protists, right? For bacteria, it's a little bit different. For prokaryotes, it's a little bit different. So yes, it is found in the nucleus, and yes, it is wound up in chromosomes. And when I say it's wound up in chromosomes, some of you might recognize this picture here. Do you see this X? Okay. This X is a chromosome, this X-looking thing. And the DNA, which you'll recognize as this coiled ladder structure, is coiled up and coiled up again into this super coiled structure that, if you stand back and look at it, kind of looks like this big crazy X. So there is just loads of DNA wrapped up in one of these things. Now, when I talk about things like genes, like the gene that I have specifically for blonde hair, it's just a small segment of a little piece of DNA. There is so much more DNA packed in there. So do we have a lot of genes? Yeah, there's a whole bunch in there. And they're different lengths, right? Like one gene might be this much DNA, another gene might be much longer. And there's padded sections in there as well that don't specifically code for something. But that's the code that makes us what we are. Now, to get back what I was saying, in eukaryotic creatures like ourselves, our DNA is wrapped up and kept safe like that in these chromosomes, in the nucleus, right? We have a nucleus and we wrap our DNA up into chromosomes. So in a prokaryotic cell, the DNA is just all loosely in there in like a big circle. So they don't have a nucleus. They won't wrap up into chromosomes, okay? Got it? Okay. So you don't need to write this down. We already discussed some of these things, but why do we care about DNA? It is a big deal. And the joke here that DNA is life, the rest is just details, is pretty close to the mark, right? It is very, very important stuff. If we don't understand DNA, we can't understand genetics. We can't understand the characteristics that you have. We can't understand how those characteristics will get passed down to your offspring or for any other creature on Earth. Sometimes we use it for forensics to identify, somebody in the case of a murder or something like that. We use it for genetic engineering. How in the world can we do something like take a gene, some piece of DNA out of a jellyfish and insert it into, say, a tobacco plant and make the tobacco plant glow in the dark? Or take the same gene and stick it in a fish and make a glow-in-the-dark fish? We do that stuff, and we do it because we understand DNA and how it works and how it's utilized by cells, and we can start to tweak it and mess with it. There's some power there, right? There's some really neat stuff we can do. Some dangerous stuff too. Treating diseases, right? Even things like curing cancer. So today, if you look at how we treat cancer today, I guarantee you in the future, quite hopefully in your lifetime, we'll look back today on today's day and age and our treatments and think like they were barbaric. In the same sense as how you might look back in the Middle Ages and think about how something like a wound was treated. If somebody got a nasty infection, like got cut on the leg in battle or something like that, and it turned all gangrenous and nasty and was really, infected, how did they treat that? They didn't have antibiotics. Yeah, they just cut that leg off. Got a red hot poker and cauterized it, burned it. They didn't know they were treating bacteria and killing the infection. They just knew they had to cut that leg off, and that was how you were going to survive. Now today you'd be like, what? Give that guy an antibiotic, right? It'd be just fine. But that's how much medicine changed. When we look back today, and today from the future, I think that many, many types of cancers you're going to see getting cured from nothing more than a pill. Like, why in the world do we have to irradiate somebody with radiation or actually cut the cancer off? Just take this pill. We'll modify the cells, modify the DNA, we'll fix it. So that stuff is happening, and it's going to be in the future. You guys can be a part of it. Vaccines, all the current vaccines rely on our knowledge of DNA and RNA. In fact, the older vaccines do as well to some degree. So this is all really important, interesting stuff. Don't write all this down. I just want to give you a little bit of background because it is kind of cool. A little bit of history. So if we roll back the clock way back, we'll hit a guy called Gregor Mendel. Do you guys remember Gregor Mendel? Did you guys learn about him in 7th and 8th grade maybe? So Gregor Mendel is an Austrian monk, and he is super infatuated with pea plants. Ringing any bells? And he starts growing these pea plants. Got a lot of time. He's a monk, and he's looking at the characteristics that the pea plants have. Some of them are tall, some are short. And he finds some that have like pure, they always breed tall no matter what, and some that always breed short no matter what. And so then he's like, hey, what if I cross these? What if I take the pollen from this one and pollinate the flowers on that one? What will I get, right? And he realizes that through this whole process, he discovers all the laws of heredity. Like, why does a plant that's crossed between a tall plant and a short plant always come out tall? But if you cross two of those, sometimes the babies come out short. So what he realizes, and the big piece here, is that he terms this stuff factors. And he realizes that these factors come in pairs, and you inherit one from each parent. So you had this tall factor and this short factor, and if your parents were tall and short, you got one from each parent. And then the babies would express the tall one over the short one. And so that's getting into dominant and recessive and all that. Some of you guys remember that stuff. But the key is without any knowledge of DNA, he realizes that there's these things that are being passed down from the parents, and he's going to call them factors. Today we call them genes, right? So, and he realizes that they're inherited from the parents. That's a big discovery. And I'm leaving a whole bunch of people out here. Jump forward to 1869, and Friedrich Michener discovers the actual molecule deoxyribonucleic acid in the nucleus. So he realizes, he discovers the chemical deoxyribose, and it gets named deoxyribonucleic acid because it's an acid, and he discovered it in the that center dark ball of the cells, of eukaryotic cells. Kind of a big deal. Now, we still don't know that that's what carries the genetic code, right? There's some folks that think they have an inkling, but there actually becomes quite the controversy that I didn't put up here about, is DNA the genetic code, or are proteins the genetic code? Because there's way more amino acids than there are bases. So those are the two building blocks of those two. So there's this huge controversy that goes on, and famous experiments that happen to actually discover that DNA is the stuff that carries the code. Okay? I put Frederick Griffith in here because he has a really interesting experiment where he discovers that there is this transforming molecule, and it can change the genetics of a creature, can change the phenotype of a creature. And all the way down to Watson and Crick, who are the two most famous names up here, who actually discovered that DNA is shaped like a double helix. So let me show you this experiment that Griffith was involved with here, because it is kind of cool. You don't need to write this down. I'm not going to expect you guys to remember it. So this wasn't so great for the mice, but he uses mice, and he has two different strains of pneumococcus. So like a bacteria that one of them is deadly and the other one is not deadly. Okay, so just think of it like that. You've got two bacteria. One bacteria will kill the mouse if he injects it in. The other bacteria doesn't kill the mouse. But the two bacteria are pretty closely related. Now, so he injects both of these, and obviously the dangerous one, the virulent one, kills the mouse. The mouse dies. The non-virulent one, the one that is not deadly, doesn't kill the mouse. Okay, everybody knew that. Then what he does is he takes and he kills, with using heat, he kills the virus, or the bacteria that is deadly to the mouse. Okay, he kills the bacteria, injects it in the mouse, and the mouse doesn't die. It's like, that's interesting. So if the bacteria is dead, it can't kill the mouse. Then he does something else that's really unique. Pretty interesting thought here. He takes the heat-killed deadly bacteria, and he injects it in the mouse. He knows it's not going to kill the mouse because it didn't before. But then he takes the bacteria that doesn't kill the mouse and injects it in there with the other bacteria that would kill the mouse. And the mouse dies. Weird. So he's like thinking about this and going, hey, there must be some special factor that is still in there, even though I killed the bacteria, that somehow the non-deadly bacteria picked up and it transformed them into deadly bacteria. And today we realize and we know that this is DNA. So even though he killed the bacteria that was deadly, that DNA from that bacteria was inside the mouse. And bacteria are phenomenal at just picking up random bits of DNA and adding them to their code. And then he took it one step further. He took a sample of that bacteria out of that dead mouse, grew it, put it in another mouse, and it killed that mouse too. These poor mice. But he went ahead and showed that, that gene, or we didn't know it was a gene at the time, that characteristic got passed down generation to generation in the bacteria after they had changed. It's really weird, right? So that was kind of a big deal. The other one, the one that I want you to know, and jot down, you don't need to know the date, but Watson and Crick, they win the Nobel Prize for discovering the actual shape of DNA. We all draw this double helix ladder looking thing today, but these are the guys that sorted out what it looks like. Imagine sorting out what DNA looks like without ever being able to see it. That's some complexity there, right? And this story of how these guys did this, it's quite the, I don't know, twists and turns of things, because they didn't do it all on their own, and they won the Nobel Prize for it. In fact, there's a lot of folks that would say that they actually stole somebody else's work to do this. And her name was Rosalind Franklin, and she was a famous crystallographer. So the way this was kind of accomplished is that a crystallographer can take X-ray pictures of a crystal. So think of something like quartz, okay, like the mineral quartz. We could shine X-rays through it and develop the film, and you're going to get this picture that kind of has these weird patterns on it of dark lines and things. And by looking at that picture, as a chemist, you work backwards to figure out how the elements are bonded together and what shape they are. So you figure out that silicon and oxygen are bonded together in this weird tetrahedral pattern to make up this mineral we call quartz. Crazy hard, complex stuff. But Rosalind Franklin is like, she's the bomb at this. She's like really good. Like, this is her field of study. And Rosalind, if I remember the story correctly, had a picture that one of her graduate students, they were researching DNA, and she had some crystallography pictures, some X-ray diffraction pictures of DNA. And there was one sitting out on her desk, and she wasn't in her office. And Watson and Crick went to visit because they had questions about, they were trying to figure out the structure of DNA, and obviously she was working on that too. And somebody let them in her office, and they went ahead and took a look at her research and things on her desk without her ever knowing anything about it, and then went back and sorted it out and won the Nobel Prize. Now, it happened after she later died, and they never awarded her any accolades or even the prize or anything like that. And now there's rules against posthumously awarding the prize, but she should have gotten a whole bunch more recognition than what they gave her. There's no doubt. But yeah, we still recognize, and they did put a lot of other work in. It isn't like they just stole the whole thing, but it's a real kind of sordid tale, right? But anyway, Watson and Crick are the big names that we remember for the straight shape of the double helix. Okay, this is one of the pictures. This is the famous photo, the infamous photo that they kind of stole from Franklin. And you can see, like, can you imagine working backwards from that and figuring out that DNA looks the way that it does? They knew which chemicals were in there. They just didn't know how they were stuck together. And they didn't know that it was twisted. And this allowed them to sort it all out. It's pretty cool. Okay, what is DNA? So I want you to jot down that it is made up of nucleotides. In a second, I'll give you a picture of a nucleotide, but I can kind of rough it out on a sketch here for you. You're going to need to get comfortable with the fact that we're drawing pictures of things that you can't see. So we can draw them real basic, we can draw them more complex, we can draw them more complex than that. So in this picture of DNA, which you'll recognize, these blue stripy things that kind of look like ribbons, this is the backbone of DNA. So by backbone, I mean it's one side of the ladder. See how there's two? If I unwound it, there'd be two. The backbone of the ladder is made up of two molecules that are the same in every single creature on Earth. And they are a sugar called deoxyribose and a phosphate group. And it just alternates. You can't see them in the picture because it's a blue ribbon, but it alternates sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Now, off of the sugars that you can't see in here hangs something called a base. It's called a base because it is basic. Right? It's sometimes referred to it as a nitrogenous base because it's made up of a bunch of nitrogens. And there's only 4 bases: adenine, thymine, guanine, cytosine. And they alternate all down as the rungs of the ladder. One hangs off one side, the other hangs off the other side, and they're bonded together. So you don't need to write all this stuff down, but just get down that it's made up of nucleotides. So a nucleotide, and I'll show you a picture here in a second, is just one phosphate, one sugar, and one base. It's a single unit of DNA. And why do we care? Because that's how this stuff is built. It's built in these little chunks called nucleotides. It's just one of each. There's 3 things in DNA, it's one of each. Cool. Okay, just get down made up of nucleotides. And yeah, it looks like a ladder, doesn't it? Especially if we straighten it out. Okay, let me show you a picture here. Go ahead and jot down what makes up a nucleotide. It was on that other page as well, but here I've underlined it specifically. A nucleotide has a phosphate group, it has a sugar called deoxyribose, and it has a nitrogenous base. And here's a picture of one. You are not going to have to remember exactly what this picture looks like perfectly, but you should be able to recognize that there's 3 parts there, right? This ball out here on the end is the phosphate group. Connected to it is the sugar. Connected to that is one of the bases, A, T, C, or G. There's only 4 of them. And the end of this base over here, these hydrogens, would be bonded out to another base on the other side of the ladder. It's made in those little chunks like that. Phosphate group, a deoxyribose, sugar, and a base. All right, don't try and draw the picture yet. I'm going to give you a picture right at the end, and it'll have everything on it for you. Okay, let's write down the bases. Right? So here are the bases. There is adenine, guanine, cytosine, and thymine. Not surprisingly, we abbreviate them AGCT. So when you hear me say the DNA code reads A, C, C, T, G, A, C, I'm just abbreviating these chemicals. Okay, looks like my words got shifted a little bit down there on the pictures, but that doesn't matter. I don't expect you to remember what the pictures look like, but notice that they are all just different chemical structures. And we've got about 3 billion of these, pairs of these, in the human genome. That is a lot of code, isn't it? You will need to know this bit up here. A's always pair up with T's, and G's always pair up with C's. Super easy to remember, though, because look at the G, see the curve? Look at the C, see the curve. They're both curvy, whereas A and T are more pointy. That's how I remember it. A's and T's and G's and C's. Because we will give you code today that will be a whole string of letters, and you'll have to make the complementary code beneath it. But it's easy. If you've got a C, a G goes under it. If you've got a G, a C goes under it. There's a T, an A. There's an A, a T. Super easy stuff. Okay. This is the one that I would like you to draw a little bit of. You don't have to draw the whole thing, But this is a great diagram. It's a simplified diagram of what it actually looks like. We've unwound a stretch of DNA, and there's two sides of the ladder, the backbones here, if you will. In going down the backbone on the right over here, I labeled a couple of these. The blue ball things I labeled phosphate, and the little hexagonal looking things here, little polygons, are deoxyribose sugar. So see how it alternates? Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. If you remember back to what we talked about bonding, there's, remember, ionic versus covalent bonds? Which ones are stronger, covalent or ionic? Covalent, fantastic. The sides of the ladder are covalently bonded, the phosphates and the sugars, because you don't want that stuff falling apart. It needs to be very, very strongly bonded. However, the center of the ladder, the bases stick together too, but they're stuck together with very weak hydrogen bonds. And the reason I specifically threw the word in there is weak, because in order to utilize your DNA, the cell needs to pull it apart. Pop, 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 pop. Two sides of the ladder. And then the cell reads one side to eventually give us a protein. So you can see look at the bonds here, these hydrogen bonds. Notice this is an important characteristic. C and G, how many bonds are there between C and G? 3. But A and T only have 2. So this is kind of partly why A always pairs up with T versus pairing up with the G, because of the nature of the bonds. So between C's and G's, there are 3. Draw 3 little dashed lines or something in there to remember. And A's and T's have 2. But your cells will pull that apart, not only to use it in order to make proteins, but also to copy it. It has to come apart. All right. You probably don't need to write down the words because I've got them on here. If you've jotted them down on there, you're good. So DNA replication. This is the last bit here that I want you to get. In order to make new cells, like when you cut yourself or you skin your knee or something like that, you have to create new skin cells. In order to do that, existing cells grow larger and divide. But when they divide, you have to have an identical copy of DNA in both cells. And this is how it's done. It's called DNA replication. You're copying the DNA. It is kind of unique how this works. The way it happens, well, first of all, it happens during part of the cell cycle called interphase. That'll be important when we talk about that here real soon. So make sure you get down that it happens during interphase, longest part of the cell cycle. You've got your old DNA here. In this picture, it's shown in red. You're going to have to color this today, so pay attention to this picture because you're going to be coloring this on the worksheet today. The red here is the old DNA. It's in the cell. It's coiled up into the double helix. It starts to unravel. There's special proteins that unravel it and pop it apart. See how the strands are popped apart? Kind of makes a V like this. So this process is called semi-conservative because of the fact that when we're done, each set of complete DNA will have one new strand and one old strand on it. Check out how it works here. After the old DNA is pulled apart, new nucleotides come in and get stuck on there. See this G stuck to the C, T to A to T, C to G, T to A, C to G, and you build a new strand on one half of it. The same thing's happening on the other side. So when it's done, these two completed pieces of DNA are going to go into 2 different cells as the cell divides. So if a cell grows and divides and replicates its DNA, which cell has the old DNA and which cell has the new DNA? Trick question. Neither. Both. Each cell has one strand of old and one strand of new, making the completed DNA molecule. Do you see it on here? That's the point I'm trying to make. Each cell, once it's divided, has one strand that's old, one strand that's new. Can you see it? It's interesting. It's unique. It's interesting how this happens, because you might think, well, we just copy all of it, and the new stuff will go to one cell, the old stuff will go to the other cell. But it's not. When you look at a cell that's divided, you can't really say, like, that's the new cell, that's the old cell. They're the same. Okay, make sure you get that jotted down. If you want to roughly draw this picture, it might be helpful, especially if you've got a colored pencil or something, to kind of just, you don't need to write in the bases, but draw the double helix there pulled apart, and then draw a new strand happening on the other one. That will probably be helpful for the test. All right, after you're done with that, that is all.